type of spermatogonia are the AD type. Okay, those spermatogonia type AD, they are the most immature, and they are the spermatogonial stem cells. Okay, they are the most immature, and they constitute the reserve population of the germ line. يعني germ cells reserve population تاعتهم هي spermatogonia type AD. يعني those are the cells that are able to proliferate, to conserve a pool of them. Okay, and those are the cells that will differentiate to give all the other cells, all the other cells necessary to produce the sperm cells. Okay, so they are the reserve population of the seminiferous tubules. It's very important to have this reserve population to be able to always produce the sperm cells. Okay, إذا ما في spermatogonia type AD, there will not be production of sperm cells. Okay, so they are the reserve cell population, and they are the most stem cells in the seminiferous tubules. Those AD spermatogonia will proliferate, will divide by mitosis to give one similar type AD spermatogonium. Okay, and by producing one similar cell or similar cells, they will be generating and conserving this pool of stem cells. Okay, and we should always conserve a pool of stem cells that is able to later proliferate and differentiate to give the mature cells. Okay, so this AD cell will divide to give one identical AD cell. Okay, and another cell which will be another cell that will proliferate and will be able to differentiate later to give the type B spermatogonia, and then the spermatocytes, and etc. Okay, and this other cell coming from the division of the AD cell is called the spermatogonium type AP, is the AP spermatogonium. Okay, so the AD spermatogonium will divide to give two cells, one identical AD cell and another AP cell. The identical AD spermatogonium will be also a reserve, yani will constitute the reserve population of the germ line, and the other AP cell can can divide and differentiate later to give the type B spermatogonia. Okay. So as I said, the type AP spermatogonia, they repeatedly divide by mitosis to produce identical clones. Here we have the AD, it gave two cells, one AD and one AP, and the AP cells will divide by mitosis to give identical cells. Yani all those cells, they are AP cells identical to this one, the first one, and all the here also we have the same representation. AD spermatogonium will divide to give one AD identical cell that will identical to the mother cell, and one AP cell. This AP cell will divide later, and an AD cell, they don't divide much. Okay, they are stem cells, they don't divide a lot, but the AP cells can divide more than the AD cells. They will divide by mitosis. They will undergo several mitotic divisions, okay, to give other AP cells. All of those are AP cells. And at certain moment, those divisions of the AP cells will stop. Once the divisions will stop, the AP cells will differentiate and will become type B spermatogonia. Okay, and now all the cells coming from one AP cell will be connected by cytoplasmic bridges. If you remember, we talked about this also, about the cytoplasmic bridges in the first chapter. So all the cells coming from the division of one AP cell will be connected to each other by cytoplasmic bridges. Why do we have those cytoplasmic bridges? Because once the mitosis occurs here, the cytoplasm will not completely divide. Yani the nuclei, we have a complete nuclear division. Sorry. but the cytoplasm will not divide completely. So the cytoplasm of the two daughter cells will remain attached to each others by those cytoplasmic bridges. Okay, and this will continue to all the steps of the spermatogenesis. Yani during all the steps of the spermatogenesis, we will have those cytoplasmic bridges that will only be disrupted when we will obtain the mature spermatozoa. So they will only be disrupted. Yani the spermatids will be also connected by those cytoplasmic bridges, and those bridges will be disrupted. Yani the spermatids will differentiate altogether to give spermatozoa. And once those spermatozoa are mature, become mature, and they are ready to be released in the lumen of the seminiferous tubules, at that moment, the cytoplasmic bridges will get disrupted. Okay. Now, all the cells connected, we said that those cytoplasmic bridges will allow for those cells, and coming from one division, for those cells and for those cells to synchronize their development and synchronize their division. Yani they will divide at the same time and will differentiate at the same time, because due to those bridges, they can exchange cytokines, growth factors, proteins, and enzymes necessary for the cell cycle progression, and so they can progress in the cell cycle in the same manner. Okay, so due to those cytoplasmic bridges, their development will be synchronized, and all the cells connected by those bridges will be called a syncytium. They will be called to form a syncytium. A syncytium, yani, it's like one cell with many nuclei. Okay, so here we have many cells, yani many connected cells, each one having a nucleus. Okay, but since the cells are open to each other, they are exchanging materials with each other, we call them a syncytium. Okay, so as I said, the AP cells will divide by mitosis. They will undergo several mitotic divisions to give the type B spermatogonia. So here we have several mitotic divisions. I didn't represent here the cytoplasmic bridges, but you should know there is cytoplasmic bridges between all of those cells. Now they undergo several mitotic divisions. We'll say X number of divisions until we will reach the type B spermatogonia, and this number of divisions the number of mitotic divisions that one AP cell will be subject to will define the number of cells in a clone. Okay, and the number of cells coming from one AP cell. Okay, so this the number of mitotic divisions until the stage of spermatogonia B. So the number of mitotic divisions until we reach the type B spermatogonia will define the number of cells that we will have in one clone. And a clone, yani the cells coming from one cell. Okay, they are called a clone because normally a clone, yani a cell identical to the mother one. لما تقولوا cloning. So once a cell will divide to give other cells, it's like it's forming a clone. Okay, so the number of mitotic divisions until the stage of type B spermatogonia will determine the number of cells in the clone. But this number, yani those cells, not all the dividing cells will remain alive. Many of them will die, and almost 2/3 of the spermatogonia will die by apoptosis. Okay, so the apoptosis will affect 2/3 of the spermatogonia. So as the number of cells in a clone will be reduced due to apoptosis, and this apoptosis will touch or will affect 2/3 of the spermatogonia.
دكتور
Yes.
هلا تاني وحدة تايب AD اللي اجت من أول AD شو دورة؟ كمان بتكفي مثلها؟
no it will يعني هول بيخلوا يضل عنده reserve population. يعني هول ال AD the initial ones okay they will divide. طب إذا هي عملت division وعطتني 2 AP cells رح يضل عندي reserve population شيء، رح يضل عندي very stem cells يلي they can be activated if there is any problem in the any cytotoxic problem that happened to the germ cell line. We will need to generate new cells that are able to form the sperm cells. If this AD spermatogonium will give 2 AP cells okay رح يضل عندي شيء أنا AD cells.
اوكي يعني renewal كأنه فينا نقول لل AD.
Yes, renewal. يعني it's يعني it will give one AD to keep this pool of reserve cells يعني as a renewal of those AD حتى يضل عندي reserve cells. Okay.
Okay, thank you doctor.
Yes, and then
راح الصوت.
ايه
معقول بعد مش هتكفي؟
بعد في ربع ساعة.
ايه بتخيل بتكفي.
أكيد حتكفي.
سؤال.
hello, sorry, I had an internet problem. I will continue. We're here. We said that the AP cells, after they divide and at certain moment they stop dividing and they differentiate to give the type B spermatogonia. Those type B spermatogonia will divide by mitosis to give the primary spermatocytes. The primary spermatocytes will undergo a growth phase before they enter in meiosis. So in the pre-leptotin stage, before the meiosis, they will undergo a growth phase. They will duplicate their DNA also. If you remember, we said before each meiosis, the cell should duplicate its DNA content, and they will increase in size. Okay, and then they will start their meiosis, the meiosis 1, to give the secondary spermatocytes, and then the secondary spermatocytes will undergo the meiosis 2 to give the spermatids. Now the primary spermatocyte, the meiosis 1, all the meiosis have a duration of 24 days, okay? And the longer meiosis is duration is for the meiosis 1. The meiosis 1 lasts for 23 days. However, the meiosis 2, which from which we will pass from secondary spermatocyte to spermatid, this meiosis 2 is lasts for only 1 day, okay? So this meiosis 1 is long, and the primary spermatocytes, they spend a long time in their prophase 1 and mostly in the Pachytene stage. In this Pachytene stage, the 50% of the primary spermatocytes will die by apoptosis also. So many of the generated here, each cell will divide to give 2 other cells, and all of them will be connected by cytoplasmic bridges, okay? So 50% of those primary spermatocytes will die by apoptosis, and this apoptosis will occur during the Pachytene stage of the meiosis 1. And the others, that those who will survive, will continue the meiosis to give the secondary spermatocyte. The secondary spermatocytes will give the spermatids. Now you know, and then the spermatids will differentiate. Each spermatid will differentiate to give a mature spermatozoa. It will change its shape, and we will talk about the changes that occurs in the spermatid to become a spermatozoa. Now, if you remember, we said that the primary spermatocytes, they are diploid cells, but after this first meiosis, they will give secondary spermatocytes, which are haploid, so having 1 set of chromosome. So they have 23 chromosomes, those secondary spermatocytes. The primary spermatocytes, they are diploid, they have 2 sets of chromosomes, they are 2n, okay? They will give by meiosis the secondary spermatocytes, which are haploid, having 23 chromosomes, but with 2 chromatids each. And as a result of the second meiosis, we will have spermatids, haploid spermatids, having 1 set of chromosome and 1 chromatid for each chromosome. So at the end, each primary spermatocyte will give rise to 4 spermatids. So each primary spermatocyte will give 4 spermatids, and the spermatids will differentiate into spermatozoa. This differentiation process, the cytodifferentiation process to pass from the spermatid to spermatozoa, is called the spermiogenesis. And we said here that the meiosis 1 lasts for 23 days, the meiosis 2 for 1 day, and the spermiogenesis lasts for 23 days, almost also for 23 days. And here in the mitosis, it will last for 16 days, almost also. So now we will move to talk about the spermiogenesis, or the cytodifferentiation of the spermatids. So the spermiogenesis is the process of differentiation that happens to the spermatids. Following this differentiation, those haploid around, or during this differentiation, those haploid round spermatids, if you can see, they are round cells. They are round spermatids. okay, they will differentiate to give spermatozoa which are more elongated and having a flagella capable of motility. Okay, so the spermatids will undergo a series of events to become spermatozoa capable of motility. The duration of the spermiogenesis is 23 days, and the haploid round spermatids undergo important morphological, biochemical, and physiological transformation. So there will be many transformation inside of those spermatids to become spermatozoa, and at the end of spermiogenesis, we will have the formation of mature gametes, which are the spermatozoa, and at the end of the spermiogenesis, we will have the spermiation, which will occur. We should be careful to differentiate between spermiation and spermiogenesis. The spermiation is the release of the spermatozoa into the lumen of the seminiferous tubules. Okay, so at the end, when the spermatozoa are produced and there will be the spermiation, they will be released in the lumen of the seminiferous tubules. So the spermatids, as we said, they will remain connected to each other by narrow cytoplasmic bridges, and those bridges will only break down upon release of the spermatozoa into the lumen of the seminiferous tubules, and this explains the synchronous development of the spermatozoa at any one part of the tubule. So as we said here, that those spermatids will remain connected even while getting differentiated. They will remain connected to each other by those cytoplasmic bridges, so they will differentiate at the same time, okay, and in the same manner, all those who are connected to each other to give mature spermatozoa, and once those spermatozoa are produced, then we will have cytoplasmic bridges will be broken, and we will have the spermiation, which is the release of the sperm cells into the lumen of the seminiferous tube. Okay. Here we can see this is a seminiferous tubule, a cross section of it. Here you have its periphery or the basal membrane. You have the type A spermatogonia here near to the periphery, and more we get differentiated, more we go inside of the seminiferous tubules. Okay, I forgot to say that the primary spermatocytes, they are the largest cells of the germ cells. Okay. So the primary spermatocytes, because they undergo a growth phase, they increase in size, they will become the largest cells. They are the biggest and largest cells of all the germ cells. Okay, and the spermatids, when the spermatocytes divide, they will give, each one will give two smaller secondary spermatocytes. So the secondary spermatocytes are smaller in size than the primary spermatocytes, and the spermatids obtained also from the secondary spermatocytes are a little bit smaller in size, and they are round shaped, and since they are differentiated, we will find them here close to the lumen, near the lumen. We can find here, you can see this big cell. In purple, you have the nucleus of the cell, and in white around it, this is the cytoplasm. This big cell is a primary spermatocyte. Look at its size. This other one also. Here, those small cells, they are the spermatids. They are close to the lumen, and then they will start to differentiate to give the sperm cells. Look at those cells having this thin nucleus, elongated nucleus. Those are differentiating spermatids. They are spermatids in differentiation, and finally they will become sperm cells like those one that will be released in the lumen.
دكتور
Yes.
هلا التايب بي بيصير قبل البرايمري سبرماتوسايت؟
Yes.
مش
The type B will divide by mitosis to give the primary spermatocytes.
يعني هو آخر stage قبل الميوسيس.
Yes, يعني هو the last cell that does not enter the meiosis. Yes.
does not enter أو does enter.
لا does not، لأنه اللي بيعملوا meiosis هن ال primary and secondary spermatocytes. كل شيء قبلهم ما بيعمل meiosis بس mitosis.
بيعمل ميتوسيس صح؟
Yes, the type B spermatogonia will divide by mitosis to give the primary spermatocyte. يعني بيعملوا
okay.
mitosis هن. Okay. And so this spermatogonial proliferation يعني or those spermatogonials هون we have the spermatogonial stage. Okay. يعني هون we will start by having spermatocytes and spermatozoa, but كل شيء before we only talk about spermatogonia and the spermatogonial phase. Okay, we talk about the spermatogonial phase. Now we'll
دكتور حنشرح بعد كتير اليوم.
عندكم شيء بعد؟
عنا لكتشر هلا واحدة ونص.
ايه هلا بعد شوي.
طيب اوكي I will stop. بس إذا
دكتور عنا لكتشر هلا بعد شوي.
okay I will stop.